Hello, my name is Oliver Payne and I'm a postdoctoral researcher working with Professor Catherine Lewis at King's College London. Today I'll be talking about polygenic scoring methods, comparing various methods to one another, and also describing resources we've developed for the calculation of polygenic scores for research and clinical purposes. I have no conflicts of interest to declare. So my presentation is split into three sections. First, I'll give a brief introduction to polygenic scoring, then describe our work comparing different polygenic scoring methods, and finally introduce an easy and reliable pipeline for polygenic scoring that we've developed called GenoPred pipe. So first, a brief introduction to polygenic scoring. A polygenic score is a way of summarizing an individual's genetic risk or propensity for a given outcome, typically calculated based on an individual's genetic variation and genome-wide association study summary statistics, referred to as GWAS SumStats. Polygenic scores are a useful research tool and could also be useful for personalized medicine as their predictive utility increases. For polygenic scoring, the GWAS summary statistics are typically processed to identify variants overlapping with the target sample, account for linkage disequilibrium between variants, and adjust the GWAS effect sizes to optimize the signal-to-noise ratio in the GWAS summary statistics. Therefore, an individual's polygenic score can vary due to the genetic variants considered and the allele frequency and linkage disequilibrium estimates used to adjust the GWAS SumStats. Often in research, these factors vary depending on the target sample, using the intersect of variance between the target sample and the GWAS, and using estimates of linkage disequilibrium and allele frequency from the target sample itself. Now, this isn't ideal, and an alternative strategy is to use a reference standardized approach, whereby a common set of variants are considered for all target samples, and the linkage and allele frequency estimates are derived using a common ancestry matched reference sample. This reference standardized approach is advantageous when using polygenic scores in both clinical and research contexts, as it allows polygenic scores to be calculated for a single individual, and it avoids variation in an individual's polygenic score due to target sample specific properties which might influence the result. Now I'll be presenting our work comparing polygenic scoring methods. This table shows the leading polygenic scoring methods as reported in the literature. The PT plus clump approach is the traditional approach whereby LD-based clumping is used to remove the linkage disequilibrium between lead variants in a GWAS, and a range of p-value thresholds are used to select the variants considered. The others are more recently developed methods that use LD estimates to jointly model the effect of genetic variants, and typically perform shrinkage to account for the different genetic architectures. Like the p-value thresholding approach, several of the newer methods apply multiple shrinkage parameters to optimize the polygenic score. When multiple parameters are used, a validation procedure such as tenfold cross-validation is required to avoid overfitting, whereby the variance explained estimate is artificially high due to trying many different p-value thresholds or shrinkage parameters. In contrast, some methods provide a pseudo-validation approach, whereby the optimal parameter is estimated based on the GWAS summary statistics alone, not requiring a validation sample. Another approach that doesn't require a validation sample is to assume an infinitesimal genetic architecture, though this approach works best for highly polygenic phenotypes. A third option is to model multiple polygenic scores based on a range of parameters, using methods such as elastic net to account for the correlation between the polygenic scores. We compared methods using a range of outcomes measured in two target samples, UK Biobank and the Twins Early Development Study referred to as TEDS. We used two samples to ensure our results were not target sample specific, and we selected the traits on the right as they have a well-powered, publicly available GWAS and represent a range of genetic architectures. As I described previously, we used the reference standardized approach when calculating the polygenic scores. LD and allele frequencies were estimated using two reference samples of differing sample size to evaluate the importance of reference sample size across methods, and these reference samples were the European subset of 1,000 genomes phase 3 and an independent random subset of 10,000 European UK Biobank participants. HapMap 3 variants were used throughout, as these variants are typically well captured by genome-wide arrays after imputation, provide good coverage of the genome, and also reduce the computation times. Several methods already provide LDMHCs, including only HapMap 3 variants for use with their software, in line with the reference standardized approach. The predictive utility of each polygenic score approach was evaluated based on the Pearson correlation between the observed outcome and predicted values. The statistical significance of differences between predicted and observed correlations was determined using the Williams test, which accounts for the correlation between predictions from each method. This figure shows the average performance of each method compared to the best PT plus clump polygenic score as identified using tenfold cross-validation. The transparent points in the figure show the results for each target phenotype separately. I'm only showing the results based on the UK Biobank target sample when using the 1,000 genomes reference, as the results were highly concordant when using TEDS or the larger reference. When comparing methods that use tenfold cross-validation to optimize parameters, shown in red, you can see that the best methods are LDPRED2, lasso sum, and PureSCS, all outperforming the PT plus clump approach, providing a 16 to 18% relative improvement in prediction. LDPRED2 showed further nominally significant improvements over lasso sum and PureSCS on average. When comparing methods that use a pseudo-validation approach or infinitesimal model, not requiring a validation sample, highlighted in blue and purple, you can see that PureSCS and DBS-LMM methods perform well, providing at least a 5% relative improvement over the other pseudo-validation methods. PureSCS is better than DBS-LMM, providing a 4% relative improvement over DBS-LMM. It's worth mentioning that SBASER's performance improved when using a larger LD reference, on average then doing as well as DBS-LMM. When comparing the pseudo-validated PureSCS performance to tenfold cross-validation results in red, the PureSCS polygenic score performs only 3% worse than the best polygenic score identified by tenfold cross-validation for any other method, and performs better than the PT plus clump approach for all phenotypes tested, highlighting the reliability of this approach. The multi-PRS approach, shown in green, which uses an elastic net model to model multiple polygenic scores based on a range of p-value thresholds or shrinkage parameters, consistently outperforms the single best polygenic score selected by tenfold cross-validation, shown in red, with the largest improvement for the PT plus clump approach of 13%. Lastly, we tested whether fitting polygenic scores across multiple methods improved prediction, and we found it did to a small degree, though the computational burden is obviously substantial because then you have to run all of the methods. In terms of runtime, these methods vary substantially. This graph shows the number of minutes to run the method on chromosome 22 alone, without any parallel computing. You can see that PureSCS and LDPRED2 take a lot longer than other methods. However, since our study, LDPRED2 developers have substantially improved the efficiency of their software, halving the runtime, moving that down to around here, just under 25 minutes for chromosome 22. It's worth noting that the time taken for PureSCS when using a single shrinkage parameter is 1/5 of the time shown here, meaning its pseudo-validation approach is actually reasonably quick. DBS-LMM is by far the fastest of the newer methods, taking just a few minutes when run genome-wide without any parallel computing, which is impressive considering how well it performed against other methods in terms of prediction. Something I wanted to highlight is that when using SBASER, I've been using the robust parameterization option available in the newest version of SBASER, as I found SBASER was not converging properly for some GWAS. Using this robust parameterization was most reliable and did not make the SBASER performance worse except for depression using the smaller 1000 genomes European reference. You can see this here in the figure. A second point is that since our published study, I've also compared PRSCS methods using GWAS summary statistics for 17 different phenotypes derived using only the UK Biobank sample, avoiding possible quality control issues that occur from large-scale meta-analyses. The results are highly concordant to the results when using the largest publicly available meta-GWAS results, which is reassuring that our findings are reliable. However, the performance of S-Base-R did improve, again highlighting that S-Base-R is more sensitive to GWAS quality than other methods. But when the quality is good, S-Base-R performs very well, appears to be the best method in this analysis. We were also interested to see whether one particular method did better when predicting across ancestries, as some methods might highlight causal genetic effects better than others. Using the same 17 GWAS I briefly mentioned on the previous slide within the UK Biobank sample alone, I've tested the performance of PRS methods across populations, so using the European GWAS but predicting in non-European subsets of UK Biobank. As you can see, the results are very similar in each population, with the best method identified in a European target sample also being the best method in a non-European target sample. So the advice for the future research regarding polygenic score methods is, for optimal prediction, we recommend modeling multiple parameters from LD-PRED2, LASSO-SUM, or PureSCS. But if you're looking for a simpler option for looking at genetic overlap perhaps, I'd recommend using the PureSCS pseudo-validation method, also referred to as its fully Bayesian method. Alternatively, if you need to compute polygenic scores for many GWAS or have limited time or computer power, then DBS-LMM is a fast and good alternative. Although S-Base-R does very well when the GWAS summary statistics quality is good, its sensitivity to that quality means it doesn't always perform well when using the largest meta-GWAS results. Okay, so now I'm going to move on to the last section of my talk, which describes our recently developed pipeline for polygenic scoring called GenoPred Pipe. So, most of the work I've just presented is contained in the study shown in the top left, where we also describe the reference standardized approach for polygenic scoring. In the study, we provide a schematic representation of this reference standardized approach shown in the figure on the right, whereby target genetic data is first imputed if it hasn't been already, then only HapMap3 variants are retained, as these are typically well imputed and provide decent coverage of the genome, and then the sample is split into ancestral superpopulations determined by comparison to the 1000 genomes phase 3 reference. And lastly, polygenic scores are then calculated based on GWAS summary statistics processed in advance using a GWAS ancestry matched reference. Now, when we were carrying out this study and writing the code for the reference standardized approach, we wanted to ensure the results were fully reproducible, and we wanted to develop a resource that other researchers could use to calculate reference standardized polygenic scores. So we used a combination of R Markdown, Git, GitHub, and GitHub pages to create a publicly available website containing a series of readable documents describing the code and results of our various studies. The website is called GenoPred, as it focuses on genotype-based prediction. And there is a QR code here if you would like to take a look. I'll now briefly show you the website. So this is the home page of the website, which shows a list of the pages available. I'll now click on the link describing how to prepare the reference data for the reference standardized approach. At the top, it provides some information and then lists the required software below that. And then it goes through each step one by one with code chunks for users to use to reproduce the results or create their own data. Now, whilst we think this is a great resource for others to see what we've done and replicate the results, You can see there are many steps and many separate code chunks to follow, making its use quite lengthy and possibly prone to user error. So I've recently written all of the steps to calculate reference standardized polygenic scores into a pipeline using SnakeMake, along with what is called a Conda environment, which will automatically download and install all of the required software, meaning it'll create fully reproducible results. The pipeline just requires 3 input files. First, a list of GWAS summary statistics that you want to use, indicating the population in which they were derived, and optionally information regarding the distribution of the phenotype in the general population, i.e. prevalence or mean and standard deviation. Then you provide a list of target samples you want polygenic scores for, with the pipeline currently accepting Plink1 binaries, so BED-BIM-FAM, or BGen files, and also the 23andMe format. Lastly, you'll need a file called config.yaml, which describes the location of the GWAS and target list files. Then actually running the whole pipeline just takes a single line of code. I provided some test data for new users to experiment with, which can be downloaded. And then step 2 here is where the pipeline is actually run. Just writing SnakeMake, then 2 parameters indicating the computational resources you want to use and that you want to use Conda environments, and then the name of the output that you want. In this example, I'm requesting the final output, which is an R Markdown report of the ancestry identification and polygenic score results for all target samples, which involves running the full pipeline. Here is an updated version of the schematic I showed you earlier, based on what is implemented in the GenoPred pipeline. The only difference is that at the end now we generate a report summarizing the results of the ancestry identification and polygenic scoring. I'll show you these reports briefly now. This is the report produced to describe the results of a single individual. First, there are some descriptives about the number of SNPs before and after imputation and the number of reference SNPs available. Then the report describes the results of the ancestry identification analysis, first showing the probability of being from each superpopulation in the 1,000 genomes reference. In this example, the individual has a probability of almost 100% of being from a European population. You can also see the individual's position on the first few reference projected principal components relative to the 1,000 genomes population I'm showing now. And the target individual in this case is that black circle. Then the individual's ancestry is broken down into more specific populations within the assigned superpopulation. In this case, showing the individual was most similar to the Great British population and individuals broadly from Northern and Western Europe. And at the bottom shows the individual's polygenic scores, in this case for body mass index and cardiovascular disease, showing the results in relative terms compared to an ancestry matched reference, and then in absolute terms for improved interpretation by considering the variance explained by the polygenic scores and the distribution of the phenotype in the general population. Then this is an example of the report produced to summarize the results for a sample of individuals. Again, starting with some descriptives about the overlap with the reference SNPs, then showing the number of individuals assigned to each superpopulation using a hardcore threshold of 50%, with the underlying probabilities shown as a histogram below. Again, you can see the position of these individuals on projected principal components compared to the 1,000 genomes reference, and then each individual is assigned to specific populations within each assigned superpopulation. I'll skip past these though and show you the polygenic scores, which are summarized at the bottom just simply using histograms to show the distribution of polygenic scores for each population and GWAS. So I'll conclude with why I think people should use this pipeline. First, it performs ancestry classification, which is really important. as non-Europeans are often discarded from studies, and as sample sizes increase, usable non-European populations can be identified and analysed. Also, it's important to consider the ancestry of an individual when calculating the polygenic score. Second, it provides reference standardized polygenic scores, which are scaled according to an ancestry matched reference and are independent of any target sample specific properties, which is useful for research and clinical prediction purposes. Third, it can efficiently implement any of the top performing polygenic scoring methods using a single line of code, saving time and reducing user error. Fourth, it's been tried and tested. I've put UK Biobank through it and other samples and compared the PRS to the observed phenotypes, assuring me that it's working as it should. Finally, it's well documented online and produces fully reproducible results, both of which are important for progressing science. I'm showing the QR code again for the GenoPred website on the right. Please do check it out if you're interested. Lastly, I'd like to thank my amazing colleagues for their help with this work, in particular Catherine for her brilliant supervision throughout. Thank you for listening.